Good morning, everyone. I would like to welcome you. I'm sorry. I would like to welcome you all to the joint Progress Emphasis workshop on the evaluation and valorization of plant genetic resources. My name is Véronique Lefebvre from INRAE, and I worked within the Progress project. And I'm pleased to start with this workshop with an introductory talk that aims at setting the scene and formulating expected outcomes of this workshop. Let's now set the scene by understanding why it is crucial to improve the management of plant genetic resources phenotypic data. As already said yesterday, currently, almost 40% of plant species are at risk of extinction, causing loss of biodiversity. And we know plants are essential for life on Earth. But climate change is already threatening plant diversity and production. We need to provide farmers with better adapted and more robust crop varieties with lower impacts on ecosystems. We know that European gene banks conserve about 2 million accessions which can be exploited in research and breeding. And to combat climate change, we need to share phenotypic data of PGRs along with their passport and genomic data. However, our heterogeneous practices and guidelines for phenotyping PGRs across gene banks hinder easy data exchange and comparison. So we consider that the solutions are within the seeds and that sustainable and science-based coordination of phenotypic data management will foster new innovations, answering our food security and facilitating the agroecological transition. The European project Progress is developing a proof of concept for a novel European research infrastructure dedicated to cataloging, describing, preserving, and enhancing plant genetic resources for food and agriculture. And the project is organized in several work packages, each addressing key aspects of plant genetic resources management, data management. These include inventory and information system, quality standards, technologies and services, governance model and communication. And I'd like to draw your attention on work package 4, which focuses on standards and protocols for evaluating and valorizing plant genetic resources. By establishing standards and protocols, it will provide a systematic approach to assess potential of plant genetic resources. And in summary, work package 4 should help to translate genetic diversity into practical solutions for agriculture. Let's now take a closer look at the work package 4 that aims to develop and disseminate crop-specific methods for evaluation based on existing standards and protocols. This work package is structured around 4 deliverables to make plant genetic resources and their knowledge available for use in order to achieve the Green Deal. The first 2 work packages include an initial version of standards and protocols for evaluating plant genetic resources and the 2 days workshop. And conclusions from 2 days will help to improve and complete the first deliverable. And this workshop will also set the groundwork for interconnecting the different phenotypic databases with EURISCO information system. Emphasis now. It's a research infrastructure that aims to develop an information system dedicated to plant genetic resources, phenotypic data. And this European infrastructure for plant phenotyping has three main actions: to develop an integrated infrastructure of instrumented facilities, to link data acquisition to data information systems and modeling, and to develop novel technologies. Both GRACE-RI and emphasis have common objectives, and they aim to promote the exploitation of plant genetic resources for sustainable agriculture, and integrating environmental, genetics, genomics, omics data help to characterize correctly the phenotype in order to accelerating the pre-breeding and breeding for sustainable agriculture. Today, we'll run 3 thematic sessions on synergy between GRACE-RI and emphasis. There will be seed phenotyping, cost-effective phenotyping, and data management. Now let me introduce EURISCO, a web-based catalog that informs on about 2 million preserved plant accession in Europe. EURISCO contains both passport and phenotypic data of about 7,000 genera and 45,000 species that were sourced from more than 400 gene banks through 43 European countries. Besides, we have more than 10 EU-funded projects and maybe even 20 funded projects that have provided a lot of genetic resources and associated knowledge on important crop species, crop families, including cereals, solanaceae, and legumes. And the challenge now is to upload the vast amount of new phenotypic data from this project into EURISCO, ensuring it remains a valuable resource for plant research and breeding. You understand that data standardization is crucial for effective use of phenotypic data. Progress has already produced 2 documents on plant phenotyping standards and gaps here. I'm sorry. Here. And these documents highlighted a number of challenges to face. We need to ensure that the phenotypic data are reproducible. We need to make phenotypic data fair. Standardization has to enable phenotypic data to be reused by machines, to ensure semantic interoperability between information systems, to enable to merge data from different phenotyping platforms to increase the statistical power of analysis, enable to compare data to understand how the interaction between genotype and environment produce phenotypes, and to speed up interconnection to integrate various phenotypic databases with EURISCO. Addressing these challenges will help to improve the usability and impact of phenotypic data for research and breeding. To standardize phenotypic data, different measures have to be taken. First, we need to assign a unique identifier to each accession. And we need also to develop a widely accepted format for phenotypic data. We know that a phenotype is a combination of a trait name that is based on crop ontology or trait ontology, a method, a clearly defined methodology and a measurement scale, and a standardized metadata for envelotyping the environment. While describing experimental metadata is relatively easy to standardize, standardizing phenotypic methods and environmental conditions is more challenging due to lab and field-specific conditions. By addressing these challenges, we can produce fair plant phenotyping data that will establish UNESCO as a trusted open access repository. And it will generate valuable knowledge for all communities in large. What are our expected outcomes from this workshop? This workshop aims to address several key questions. How can breeders, farmers, and citizens gain easy access to PGRs and use them to speed up the green transition? What standards should be improved in Genebank inventories? What is the potential of novel phenomics technologies and their inclusion in phenotypic databases? How can we integrate different initiatives such as Progress and Passes and UNESCO? How can breeders, farmers, and citizens contribute to conservation and phenotyping? And what is needed to merge experiments from pre-existing databases to assess the effect of interaction between genotype and environment? Addressing these questions will help to define priorities to leveraging PGRs for sustainable agriculture. Here is today's programs, and I want to stress that there will be 3 thematic sessions. Each theme will feature 4 invited speakers and a common Q&A session. For online attendees, use the Q&A icon to ask questions. And for in-person attendees, we will take a few questions here in the room. And we ask you all to fit the program timings. And at the end of the day, we will try to give you a wrap-up and conclusions for the first show. I thank you for your attention. Patrick? Yes, there is one question from François. Ah, here.
No, but perhaps there are
No, François, please ask the question.
Is there a microphone?
Oh,
I shall.
sorry.
You have to wait.
Thank you.
Yes, also from infrastructure. Doesn't work? I've not assigned a unique identifier for each accession.
Yes.
Could I share my screen now?
European
Yes.
database.
So I stop sharing.
There's huge problems. Because
are you
actually each
Now I'm sharing
accession
the PowerPoint.
variety I had especially yesterday. Identifier. So
Yeah.
we get that.
Now
I'm going to tell you, okay, we will give this, but we cannot do that now. So have you got any advice on what to do in this context?
Not really advices for this. Maybe we will discuss. Not really advices for this question, but probably we will discuss during the day how to give a common, a unique accession identifier. I know that at INRAE, URGI is involved in this task, at least through the AgroDIF project. And I consider it's really important to have a unique identifier, because when we compare for instance, the results from publication for same accession. You have different names. You have the INRAE, the G2P sol name, the IPKA, the USDA name, and so on. So it's terrible, and it do not facilitate merging of data.
Okay, so you see on the screen, and hopefully also you can see it in the online for the online audience. So we are trying today to be a little bit more consistent with the Slido, using Slido to interact also with the online participants. So I am starting our first poll of the day. We'll give you let's see which one is it. The first poll of the day. And if you can use the QR code, or you just go to slido.com and put in the meeting number 22 45 36 5, and then you can vote in the poll that we have set up today as a first one to inform also our our outcomes. And the question: as a breeder or researcher, do you trust in phenotypic data collected by citizens or farmers? And we'll give it like maybe 2 minutes to see how many of these questions. We have 43 respondents so far. Maybe we'll stop when we get to 50. So 5 more. Okay. So it seems there's a little confidence in phenotypic data collected by citizens or farmers. Thank you very much for attending this first poll. We'll continue with the next presentation. Yeah.
So let's begin with the presentation, and I will now hand over the first speaker of the day, Roland Pieruschka, who will give you a more detailed presentation of the inventories results in infrastructure. And yes, it's always
Thanks a lot, Véronique. What I would like to talk about is emphasis. There are some redundancies with the presentations from yesterday for people who were there yesterday, but there are also some new things. But I think we also have a number of new people that joined today at the workshop, so it might be useful to get a general overview. Maybe also start when there is a little confidence in citizen science. I hope there is more confidence in research infrastructures. Where you jointly try to build actually 2 research infrastructure, which is good, and developing this confidence. But anyway, so let me start with, so Véronique already showed this triangle in plant, so basically phenotype, environment, and genotype that we need to understand to advance. So basically nature, nurture, to shapes that shapes the phenotype, and genomics, genetics, this is well advanced. So in plant phenotyping, this quantitative assessment of a trait and environment, this is still a lack. In the end, you can imagine the genotyping, you just use the sequence of your genetic material. For phenotyping, you have many different traits, many different environmental conditions, and developing, measuring them under all those conditions is really a challenge. And we kind of need to work together, and there is quite some demand for this to address all those different crops under diverse conditions. but also, so what is needed specifically is this infrastructure, so specialized infrastructure to characterize those traits, but also characterize the environment, so simulation and environmental monitoring, so basically envirotyping. Widely interdisciplinary approaches are needed with technology, but also expertise needed from many different disciplines. And one of the reasons to build a research infrastructure is that we actually want to work together and advance as a community, as a European community. The same goes for INRAE, as for emphasis that we want to advance Europe in those research areas jointly. So the demand for plant phenotyping, it's also relevant for breeding. So breeding was addressed yesterday a couple of times, and what we also quite often, so basically one of the goals where phenotyping is needed is in this breeding cycle. It was introduced also yesterday, where we characterize genetic diversity, establish breeding populations, select varieties, confirm the traits, and basically close the cycle. And in the sense, we need controlled environment facilities, we need field experiments. So quantitative screening in the sense is very important, but we also need to develop this technology, methods, standards that we really address the throughput that's needed to advance, to deliver what is needed to also for breeding applications. In this sense, we try to develop with an emphasis kind of a integrated concept where we have, this is a different description of this emphasis mission that also Véronique already described. So basically what we have instrumented facilities, we have this data acquisition, information systems and modeling, and in the end also knowledge technology transfer. What I added here is it's also a loop back. So this is on the one hand knowledge technology transfer. It can go towards industry to develop new tools, to develop new startup companies, basically for all those applied aspects. But there is also a substantial feedback loop, so that all this technology goes back into these instrumented facilities and basically advances on these facilities. And we have also dedicated projects that specifically address this aspect. Based on that, we also developed categories, phenotyping categories on the control field environment modeling and data management that fit into this structure and are also complementary. To give you a very short description what we mean with those different infrastructure categories. So we have controlled environment facilities. So it's mostly greenhouses, growth chambers where you can simulate the environment quite well. You have a very high level of automation. You could do deep phenotyping and you have a substantial throughput. Those are pictures actually from Montpellier, from Jülich, from colleagues in the UK. Sorry that I did not put the names of these facilities, but it's just to give you a general impression how they look like and what we actually do and how we address this high throughput. Going into the field, so we have intensive field where we, on the one hand, you have high level of basically heavy equipment where you can want to measure with a high level of precision, but where you also manipulate the environment, either with rainout shelters. You have phase facilities where you can monitor or basically simulate and elevated CO2 under field conditions over large areas, basically simulate future environmental conditions. Next to that, we have also lean field. Lean field, it's more or less rather simple field sites. They can be combined into a network of field sites. They are pretty close to breeders' applications, so rather simple equipment, drones with simple cameras. But you have this advantage of linking them, being able to link them into a networks of field sites and do experiments for extended areas. This is complemented by modeling. There is a number of different models in this graph up there. You can see that on the x-axis that you can really read those crops, and on the y-axis there are a number of different models that are available. Many of those models are in this platform here, in this black box. So it's on the website as an open source accessible. And an example is OpenSimRoot, which we are developing in Jülich, and this is also in Jülich, but also with colleagues in I think in France as well. Moving on, So this is complemented also by data management. In the end, we produce a lot of data, hundreds, thousands, if not millions of images, and those images need to be analyzed, but also used for many different, basically, this data reused for different purposes. So we try to, on the one hand, there are semantic description of data, but also metadata in this approach called MyAP, but also technical data integration and sharing. There are local infrastructure that were developed in many different countries to organize those data. And this is kind of a parallel development where the goal is also to link those national databases or databases in different institutions. And currently FAIR there is one of the important tools that we use to integrate those data. And I'm pretty sure that also Cyril will talk later on about also FAIR there. So moving on then, so the idea is that we have this 5 different infrastructure and combine them into basically into services to provide services. Those services are currently under development, but in principle there would be access, development, further advancement of practices, phenotyping practices, engagement of industry, data management, modeling, communication, training. Those are things that would need to be part of the services of emphasis. But as mentioned, it's still under development. In this sense, we also try, talking about emphasis or phenotyping as a tool, we also try to extend basically widen the scope of this, how those tools can be used. And to a large extent, we are involved in agroecology in a project called AgroServe, where there are 12 different research infrastructures. So it's really huge. There are 73 different partners. There are 140 different services. There is a substantial budget, toolbox of services that covers all relevant ecological, basically climatic regions in Europe, and those are accessible for transnational access. So basically, we had the first call that was already closed with 101 applications, and the second call opened yesterday. Yesterday, I already talked with the colleagues, and they asked about this access, and in the end, it's rather straightforward. So there is an application. The application is pretty straightforward. Important in this context is that we really talk with people running those services, because in the end, when you apply for access, and the access works in a way, you submit an application, there are independent reviewers, and you can do this access entirely for free. So those costs for this access are covered from this project, and this provides a lot of opportunities to use phenotyping facilities to, in the end, for your specific question. But to mention once again, talking with the facility operators, people who run those facilities, it's really important to, in the end, develop projects, proposals that are feasible and work and provide also benefit and also provide also output from those activities. In parallel to agroecology, there is also a project called Microbes for Climate. So this is the approach is complementary to AgroServe. It addresses basically microbes, soil, plant, environment, and there are also 140 different services. So it's very complementary, but the focus is more plant microbes interaction, where you can also apply and use the services, and the call will be open most likely towards the end of 2024. In parallel also to providing access, we also are trying to developing basically next generation tools. This is in a project called FENET, where the idea is where 4 different research infrastructures work together to basically unify their approaches to develop next generation tools, novel tools, and deliver services, new service for the research infrastructures. And actually we have Bertrand Müller sitting here. He's the coordinator of this project. If you have any further questions, feel free to talk also with Bertrand to give you some more details about this. So the idea is to develop, integrate low-cost AI-based automated solutions. So basically hardware, software, models, but also this analyzing of data. So capture, analyze large diversity of traits. So on the one hand traits, but also environmental variables, so envirotyping, and in the end enable predictions in agroecosystems. So this is kind of the vision, the mission that we have. And this is tested in a very practical, realistic scenarios that address really scientific questions. So currently burning scientific questions. This is the approach that we not only develop tools, but test them, specifically for soil, root, but also G by E interactions for cereals, but also orchards, completely different approaches that are needed. Biotic interaction, plant health, so pathogens, there's a number of pathogens involved in this use case, but also intercropping, so plant-plant interactions. And we also try to scale up, go across different scales towards farmers, but also landscape approaches. Those tools are there, more or less the development is kind of addressed towards those different use cases, but I'm looking at those different tools that we want to develop. They have quite a lot of potential to be also applied towards plant genetic resources. So it provides also a lot of room for future discussions to address these applications for also in cooperation with RACE project. Moving on, something that I wanted to very briefly address, there is also IPPN. So I mentioned at the beginning, competition is growing, but there is also a lot of collaboration partners. So it's not only competition, but we have also this tool, which is called IPPN, International Plant Phenotyping Network, which is an association, really a network across the globe where we can talk, interact with many different partners across the globe. And one of the key tools is this conference, which is the IPPS, International Plant Phenotyping Symposium. This conference takes place every 2 years. The next one will be this October in the US, and it provides opportunities. So not only think about our people in the US, Australia as competitors, but as partners and basically to talk and interact with them. And you are very welcome to join this meeting, submit an abstract, and hope to discuss, learn more about plant phenotyping. And as such, thanks a lot for your attention. There is a major thank to being in Brussels to the funders from the Commission, and happy to take questions now or throughout the day. Thanks a lot.
Okay, thank you very much, Roland. We have time for maybe one question here, and we will continue during the Q&A session. But if someone has a burning question, yeah, please. Can you speak maybe in front here or very loud? You have a mic? Okay.
Thank you. So, Formula from France, INRAE. So, thank you for mentioning Planet. Obviously, that's a important project in that perspective. But I also wanted to mention the INVITE project H2020 innovation invite testing. So, I quite believe that there are some opportunities to join forces towards innovation invite testing in the future, leveraging phenotyping technology. So, that's part of the thing we would have in our perspective.
Yes, so I don't know where the impact project, but I know it by the small windows of Toledo, because the material cost is working in it. And it's true that there will be a lot of genetic data through this project, combined with also some genomic data. And it could be very interesting to include it discuss with them how they will manage all their data to integrate to other databases. Okay, thank you very much. And now I'm pleased to welcome Jaime Prohens from UPV, University at Valencia, that will present a talk on how to make Genebank materials attractive for users and what are the Genebank perspectives. Thank you.
Thank you, Véronique. Thanks also to the organizers for inviting me to give this talk. And I will give my view on how can we make gene banks more attractive for users. Well, first is that I'm of on both sides of the table. I'm a user. I have been researcher and breeder for more than 30 years, but also have been involved quite a bit in the Genebank because I have been director of the institute that holds the UPB Germplant Bank for 8.5 years. And since 9 months ago, I'm also director of the UPB Germplant Bank after my colleague Maria Jose retired. So I inherited this challenging task. Well, to put you also into context, our Germplant Bank is small. We have a little bit less than 14,000 accessions, and we are one of the around 2,000 gene banks around the world and around 400 in Europe. Well, it's not needed to stress the importance of gene banks here, but they are essential in maintaining the food security for the long conservation of genetic resources of crops and wild relatives, and they also characterize the genetic resources at different layers. Ideally, they have to properly document the information of genetic resources and make it available, provide the genetic diversity for users, and this information must be of the highest possible quality and abiding to the applicable laws and regulations. And of course, they provide the raw material for genetic advances in yield and sustainable agriculture. There is a study that I think was commissioned by Euroseeds about the value of plant breeding in the EU in the last 20 years. Well, the genetic resources, together with plant breeding, have contributed to an annual yield growth of 1.16% every year. So it's quite a lot. And this, the plant breeding based on the genetic resources has avoided an extra emission of greenhouse gases of almost 4 billion tons, which is the whole emissions of the European Union of Europe in a single year. So it's quite a lot, the contribution to sustainable agriculture. What are the main challenges for gene banks? One of them is funding. Funding is a crucial issue. There is a disparity in the availability of funds among germplant banks. Here in Europe, we have some germplant banks that are quite well funded, and others that are striving to survive and to just maintain the collections in the cold chambers. So this deficient funding often results in inappropriate germplant management, poor quality germplant, and associated information. Here, a big issue is that the funding of gene banks depends mostly from the national level, and this results in disparities of strategies, irregular funding, depends on political will, and so on. And this difficulties the efficient coordination at the European level. Also, very importantly, the genetic resources went from a heritage of mankind previous to 1992 to resources that are under national sovereignty. So this has changed a lot the legislation and the difficulties to access to genetic resources. Before 1992, you could go, collect freely, change freely. Now the thing is more complicated. So this has made that the access to genetic diversity has become more bureaucratic, and even there are seed companies that are doing or are establishing their own private germplant banks. Why? Because accessing the public germplant bank sometimes is really complicated to get the material with all the regulations that are existing. One way to avoid these cumbersome, burdensome regulations is the multilateral system of the international treaty. However, this is restricted to 35 crops and 29 forage species. So nowadays, from 2 million accessions available in Eurisco, a little bit more than 20% are in this multilateral system, in which a change of materials is facilitated. Here we can see from two of the main germplant banks in Europe what has been the trend. Yesterday, Theo already presented for CGN, so we see an increase in the supply of germplasm. Then there is a peak around 2012, 2014, and then a decrease. With IPK, it's the same. These probably are two of the best germplasm banks in Europe. In smaller germplasm banks, the situation has been even worse than that. So the reality is that there is a decrease in the use of genetic diversity available in germplasm banks. So what can we do to improve accessibility? First, the germplasm, we have to improve the access to the germplasm that should be viable, because sometimes the materials that are sent are not viable or have low viability, disease-free, and with acceptable legal conditions. Sometimes the users say, okay, I cannot accept this legal condition, but because they are too restrictive and it's not possible to provide them. Also, the germplasm is most useful when distributed together with relevant and high quality information. There are some databases that already provide some of this high quality information. And there has to be legal certainty for both the germplasm bank and the users, and easy and transparent access to the genetic resources, which many times, especially with small germplasm banks, is not so clear. So also for improving this accessibility, we have to have comprehensive and accurate documentation of gene bank materials. Sometimes when germplasm is provided from a gene bank, it doesn't even correspond to the species or to the taxon they say in the passport data. So, and for the rest of the documentation, sometimes it's absolutely irrelevant or does not exist. Also facilitate the burdensome paperwork, including phytosanitary aspects, which are becoming more and more complicated, not only the legal transmission of germplasm, but also the phytosanitary issues to facilitate providing germplasm. There must be a fast and straightforward process for supplying germplasm to breeders and seed companies. I think that it's not acceptable to have to wait 4 or 5 weeks or months to get seeds. Also, one way to enhance attractiveness, at least in Europe, is to designate more accessions as part of the European Gene Bank integrated system. These are accessions that are designated by the germplasm banks and that ensure the long-term conservation of the European collection according to FAO and crop-specific agreed standards, that it is ensured a safety duplication in agreed conditions, that there is a record public domain accession level information through UNESCO and the national inventory system, and which have facilitated access and availability. But only around 3.5% of the European accesses now are under this European Gene Bank integrated system umbrella. Also, adapting standards like certification and auditing. For example, CGN was presented yesterday, has a quality management system. Very few banks have this. So they cannot guarantee the quality of the materials that they are providing. Clear rules on providing accessions. What's the maximum number of accessions? What's the cost, if any, of these accessions? The time required for sending germplasm. So many times this is not available for the users. Of course, including the information in databases and online platforms, such as UNESCO or Genesis and others like Germinate, or specific of crops like Jupiter, Harnestone, or the Tomato Genetic Resources Center. If the information is not available, the attractivity of the germplasm is much less. One very efficient way to promote utilization is developing partnerships with researchers, breeders, and farmers to encourage the use of germplasm materials. One way is collaborating in regeneration and phenotypic or molecular characterization and evaluation with breeding companies. Because this facilitates the selection of the germplasm by the companies. They also regenerate and can provide data on characterization and evaluation. But of course, breeding companies do not want the effort to be used by other breeding companies, at least immediately. So many times this functions with an embargo period, which usually is 2-3 years. Of course, having images and high throughput phenomic data, as had been presented before, in addition to standardized primary characterization, would be very useful and add a lot of value to the germ plan. One way to enhance attractiveness, my point of view, is a specific specialization in certain crops. I think that working on tomato, the most successful and useful germ plan bank in the world for tomato has been the Charles Reid Tomato Genetic Resource Center. It's the reference, and everybody goes to this germ plan bank when they need a tomato. They provide a lot of information, and now, unfortunately, due to this phytosanitary regulation, they cannot send material to you. So they tell you, ask if any germ plan bank in Europe has this accession you have requested. Also, of course, having high throughput genotyping available for gene bank accessions would be very helpful to increase attractiveness. This provides data on fingerprinting, genetic distances, for GWAS analysis, identification of duplicates, gaps in the collection, heterozygosity estimates, genomic prediction can help on this, taxonomic identification, and many other applications of the high throughput genotype. Well, some innovative approaches to increase attractiveness, so many of them are already in force in some germ plan banks, are providing training and support to potential users how to access and use the gene bank materials. Open days and field days for breeders, researchers, and farmers would also be a very good way to show and to increase the interest by the users. And here there is a very controversial issue. Should the gene banks be a source of breeding and experimental populations? From my point of view as user, is yes, that would be very interesting. From my point of view of germ bank manager, it's wow. Now we have to conserve hundreds or thousands of lines of magic populations, introgression lines, mutant populations, mutant collections. So this is a very big challenge, but it's clear that it would increase a lot the usefulness and the interest of the germ plant banks. Of course, improving the collections, filling the genetic and geographic gaps and of underrepresented crops and crop wild relatives. However, this is becoming increasingly complicated. It's not as before that you could go and collect everywhere. Now it's challenging, and there are many countries that they don't know how to manage this, and so as they don't know how to manage, the answer is no. And also for enhancing attractiveness, I think that policy and advocacy actions like creating progress, of course, is a way of enhancing this attractiveness, and also advocating for supportive policies and increased funding for gene banks at national and international levels. And if this could be possible, it's beyond our scope, extending the multilateral system to all crops. This would facilitate a lot, but it's a political decision. Raising awareness about the importance of gene banks through educational campaigns and outreach programs would also help in this attractive. Perhaps here, as was presented yesterday and today by Roberto, involving citizens in this characterization or evaluation of materials, or even multiplication could be useful, with many challenges, of course. Participation of private sector representatives in gene bank advisory committees would also help in this attractiveness, and establishing of partnerships with term agreements between gene banks and breeding companies and associations for providing mutual services. There are some examples of this also, like there will be a talk just after mine about the European Evaluation Network, the joint participation of as many as possible germplasm banks and collections in this EVA project would be very efficient, and extend the concept of the European Gene Bank integrated system to more accessions. As I said, only 65,000 accessions of the 2 million are under this umbrella, which is very low. A big problem. What if we are too attractive? But if you don't receive applications, the users cannot complain. But if you receive applications and you are unable to supply it, there is a problem. So it being too attractive may result in too many applications that in quite a number of germplasm banks, especially small, cannot be managed in a sustainable way without extra resources. So this is something that has to be taken into account. I think that the key is more funding and more resources for the germplasm banks. Well, in summary, as you know, gene banks are essential for food security and sustainable agriculture. There are many funding disparities and bureaucratic hurdles that impact the gene bank effectiveness. Increased gene bank attractiveness for users requires improving accessibility and enhancing the information provided on germplasm. Improving the current databases to provide extended information, including phenomics and characterization data, would increase the attractiveness. It's important to advocate for supporting policies and raising awareness, and the development of the GRACE research infrastructure will represent a dramatic improvement on attractiveness of germplasm to users. Call to action is continue with improving the gene bank function in procedures and standards, enhancing the capability of databases, facilitate as much as possible the bureaucratic and phytosanitary paperwork, engage in raising awareness among the public and among decision makers, and support actively the GRACE research infrastructure. So that's my talk so far, and well, I'm happy.
Okay, thank you very much for Jaime. Thank you very, very much, Jaime. We have one question by through the Q&A and one question in the room.
Okay.
Who starts?
Maybe I'll start with Nigel's question.
Okay, yeah,
online, because he was already there. So his question for you is, should the gene bank serve all users' requests or just professional users, or excluding farmers and gardeners?
Well, nowadays this depends on the policy of the germplasm banks. In our case, we provide to all users, including farmers, because many times farmers are interested in recovering the old traditional varieties that already do not exist in their area of cultivation. But also attending public petitions and gardeners is a way to raise awareness and also to get them involved in the importance of germplasm banks. But I understand that some germplasm banks cannot attend this type of petitions because this would mean too many petitions and depleting the stocks. But I think it's interesting to provide also to the public.
Hi, Jaime. Thanks for the excellent talk. So regarding the question by Nigel, do you think that maybe seed conservation networks would have a role in distributing to farmers and gardens?
Yes. Yeah, I think, I don't know, but in other countries, but in my experience, there has been a little bit of disconnection between germplasm banks and seed networks. And I think this has to be strengthened because they can be very complementary on the activities they perform.
And the second, more common than question about the problem of becoming too attractive. I think that one of the possible roles of GRACE-RI is, as we have seen in previous EU projects, is to generate core collections of very well characterized genetic material that represents the worldwide diversity. This way, probably a lot of requests would focus on this material, thus alleviating the need for the gene banks to distribute everything they have. So it would focus a lot. Do you think this would be helpful?
This would be very helpful because it would be a selected, understand that this could be under the umbrella of IHS, the European Gene Bank integrated system, with a duplication in other places, with well-characterized materials, both at the phenotypic and genetic level, that represent the diversity. I think this would be a very good action by the GRACE-RI.
Yes, one more question.
Okay, just
I'm sorry.
Just one question from Carla, but it's a question without answer. It's just to think.
Yes.
And after we go to another speaker.
Okay, thank you. I know we are run on time, but your talk is about how to make resources attractive. And you clearly show that in the last decade, resources are becoming less attractive. So what's happening?
Well, one of the main issues are the legal issues.
Nagoya.
Yeah, Nagoya Protocol and the MTAs and all this kind of thing. And the problem is that many times the providers, the small gene bank providers, they do not have the legal advising about how to proceed in data. They get blocked.
Is a thing, right?
Yeah.
Okay, thank you. We will continue the discussion during the lunch and the coffee break and the lunch. And now I'm pleased to welcome Sandra Goritschnig, that will present the European projects contributing to improve gene bank inventories, and with 2 examples of HN and EVA. Thank you, Sandra.
Okay. All right, I will try to be quick also in the interest of time. We started a bit late. First of all, I wanted to highlight that we're working actively to increase the participation in EGIs within ECPGR. And at the moment, we already have 120,000 accessions. So there is it's growing, but of course, there's still a lot to be done. So I showed this slide yesterday. So again, it's just maybe to reiterate how important the work of gene banks is. And probably after Jaime's talk, I don't need to emphasize so much on the importance of documentation and to understand the need that gene bank accessions are available to farmers and breeders. And the thing is that breeders, they really need to understand how do they know which accessions are interesting for your breeding project. And 2 issues that Jaime also mentioned is like in some cases, passport data on PGR is not always consistently high quality, and characterization data is also not always available. And if it has been collected over regeneration cycles, it is not necessarily digitized and available. What is important is to really connect the conservation and use of PGR through high quality documentation. And so these 2 projects that I'm presenting here as examples of all the other European and bilateral projects that are out there, just because I'm also involved in them, is really to work on improving gene bank inventories and access to PGR through different approaches. The activated gene bank network, or agent, is creating a network of gene banks to build capacity and create what we call bioresources centers. And the EVA networks are public-private partnerships that evaluate gene and materials in multi-location trials across Europe. I'll start with agent, and this is just to highlight the facts and goals. So agent is combining gene banks and bioinformatics centers. We have 18 partners and their gene banks and bioinformatics centers that together work on improving the data that is available in a fair, according to fair principles. And Cyril will talk a bit more about how we do this in order to unlock existing data sets and to also generate new data on gene bank materials, and to also work on implementing quality control and quality management principles to the data and to the conservation efforts. Agent is centered around EURISCO, where bioinformatics centers are providing or developing and providing bioinformatics tools that help the gene banks that are signified by this green circle to use these tools to better manage their collections and to better manage the data on their collections in order to provide the material and the associated data to the stakeholders that are in the outermost circle, exemplified here by the EVA network, for example, but also other researchers, breeders, also policymakers, and the general public. What we aim to do is, or to get out of this, is to create novel collaborative networks of these gene banks that provide more intuitive ways into accessing data on genetic resources to allow a better informed selection of accessions in the collections, and also to use the data that we collect from the different gene banks to do comparative analyses. So we are using wheat and barley in this project, but these concepts that are developed can easily also be applied to other crops and other gene banks. The agent gene banks have created collections of wheat and barley for phenotyping and genotyping. There are 3 different types of collections. The precision collection is made up of unique accessions from each gene bank that they have selected among their stocks, and that is in total about 12,000 accessions of wheat and barley. They've also assembled a bridging collections that identifies potential duplicates between the different gene banks, and both of these collections are genotyped and phenotyped with for some agronomic traits that I've listed here: plant height, flowering time, and 1,000 kernel weight, to assess, assess some of their characteristics. There's also a collection of standard checks. These are commercial varieties, 200, that have been phenotyped by all participating gene banks in Europe, and have created the ability to look at the environmental effect and design sort of mega environments that are providing comparable environmental conditions. The collections are also phenotyped for important biotic and abiotic stresses to collect data for genomic association and prediction. We've been doing fungal diseases in various locations, and also abiotic stress trials for drought in select locations where this is more common around southern Europe. An important, or really, in my opinion, a really interesting aspect of the agent project is also that the gene banks went into the archives and looked at historic data from their collections, from regenerations that they did during the last decades. And we found data basically going back to the 1940s, especially from IPK and Agroscope in Switzerland. And during regeneration of wheat and barley, the main traits again are plant height, flowering time, and 1,000 kernel weight. some gene banks also have collected other data. And this all has been digitized, which allows them to do various analyses such as heritability, but also genome association and genomic prediction. And if you look at the time frame of the data that's been collected, we can also obviously look at the effect of climate change on the collections. The agent tools for gene bank management that the bioinformatics teams have developed, and Cyril, I think, will be talking a little bit more about that, are also very important to really help the gene banks better manage their collections and the associated data. So we have developed data management guidelines for the phenotypic and genotypic data, standard templates for data collection and data management, curation guides and validation tools for data to make sure that they are fair and actually also reusable. We have developed an agent data portal, which is you can see maybe on the right, and which will provide all the collected phenotypic data and also historic data in a web portal, and that should go live very soon, actually. And then also importantly, data analysis tools and pipelines have also been developed in this project. And the last important aspect of the agent project is the stakeholder engagement. And this is where we also have the link with the EVA network that has been mentioned, that I'll be talking a bit more in detail, where we take some accessions from these agent precision collections and put them in the fields together with our EVA partners in various locations across Europe to look at their performance. We also interact here with the Rete Semi-Rurali, and Ricardo Bocci will be talking a bit more about on-farm phenotyping later to test this approach also in an on-farm setting. The European evaluation networks have been established in 2019. It's an ECPGR initiative, and it aims to promote the use of germplasm diversity in research breeding and cultivation. We also, in light of EGIS, we promote the exchange of PGR material with SMTA, and we do this for all accessions, irrespective of whether they're in the multilateral system or not, or in the Annex 1 of the international treaty. We want to foster cooperation between the public and private sector and involve also wider groups of stakeholders in using the PGRFA. So we have signed this memorandum of understanding that was also mentioned yesterday with ESA, or now Euroseeds, to really be able to connect with the private sector breeding companies in this respect. We are generating standardized multi-location data, and this should hopefully help us to also identify climate resilient breeding material that could be used then in further breeding programs. But in general, we really want to increase the knowledge and improve the information associated with accessions in EURISCO through also the continuous work of self-sustaining networks that evaluate the accessions in the gene banks in continuous evaluation cycles. At this moment, we are running 6 European evaluation networks. They're crop specific, and we have 3 for vegetables: carrot, lettuce, and pepper, and 2 for main field crops: wheat and barley and maize. And the newest addition is the EVA Legumes network, which has started in 2024. We are connecting more than 130 partners in those different networks, and obviously also in different combinations. And this includes more than 50 breeding companies and more than 50 research institutes and 30 pure gene banks, if you will, from 34 countries across Europe. So because this is about phenotyping, I'll really focus on like two aspects of the circular evaluation process in EVA. And that is, first of all, the standard protocols for traits of interest. It's really important to use the same protocol if you do like 10 different trials in different countries in Europe. And then, you know, it always depends a little bit on who is doing the trial, who is doing the scoring, to be able to compare the data. We're collecting data for morphological traits, agronomical traits, quality traits, most importantly for breeders, and we select the traits of interest mostly with consultation with breeders. Biotic stresses and abiotic stresses are obviously very important. Our standard protocols combine descriptors, morphological descriptors from INRAE, for example, also very published protocols from the literature, and we also obviously rely on the expertise of the various partners. We do multi-location trials in field, greenhouse, and lab to look at the behavior of the crops in different environments to see if we identify locally adapted accessions. On the right in the map, you see the trial locations of our 5 older networks, and that's more than 100 across Europe. So in most of them with the wheat and barley network. And on the left, I am showing some data from our carrot network, which has evaluated accessions for bolting in different locations. And if you see the spikes on the right, it's basically accessions that are tending to bolt, but not in all locations. So it's quite interesting to see the flowering behavior of carrot, which is an important trait for carrot breeders in different locations. We have collected more than 530,000 evaluation data points and inserted them into a Eurisco EVA intranet information system that is compatible with Eurisco and allows that after the embargo period, we can transfer this data into the public domain and make it available linked to the accessions that the data has actually come from. We have looked at more than 5,000 accessions for the different crops. We have evaluated more than 230 traits, different traits, depending on the crops. Again, we have, for example, for carrot, evaluated, more than 100 different traits. And we have collected data from almost 400 EVA trials, the majority again for the wheat and barley network. Another interesting or very important aspect is to use the phenotypic data we collect in association with the genotypic data. So we make sure that all the accessions that we phenotype are also genotyped, because it allows us to also apply a genome-wide association. Here in this example, in lettuce, we have very nice associations for 4 different traits: seed color, leaf color, and bolting time. So what I hope I have shown you now is that there are many many initiatives that improve documentation and gene bank inventories that will enable access to PGR. What is important is we need to harmonize documentation of passport data on PGR and in terms of quality across Europe. Make sure that we standardize also characterization data through the use of for example, MIAPE compliant metadata. And again, Cyril will talk more about that. And I think it's also very important to mobilize the data sets that have been created, that are existing from previous projects and also from historic regenerations, as you saw in the agent example, to really enable exploitation and use of that data with the accessions. And yeah, so perhaps Progress can be a path to implementing the results, the approaches from projects such as Agent and EVA through coordinated actions in a research infrastructure. So my acknowledgements are to all the EVA partners. I cannot put the logos because there are too many. So if you see a bit in the word cloud, EuroSeeds has been very important to connect us with the private sector, and that is really great. Of course, also the agent partners, and most importantly, also the funding. The German Ministry of Agriculture is instrumental in getting EVA off the ground and sustaining EVA. And of course, the agent project is a Horizon project. So thank you very much.
Thank you, Sandra. Is there any question in the room here? Or can you have a look on the question on my list?
Yes, there's one question.
Can you ask your question for you and answer it? Yes. I'm sorry. Can we use the genotypic and phenotypic data along with, I'm sorry. Can we use the genotypic and phenotypic data along with Jean-Claude succession? Will you share the data?
Of course, the intention is to make the data available and fair. As I said, we're both in Agent and EVA, we're using Eurisco as a central repository. And this is for the moment also only made for phenotypic data. Genotypic data has to be managed in a different way because it's just too big. But yes, we are making that available, and yeah, with the accessions. Awesome. Thank you.
Maybe I have a comment. You say that you are. For maybe several speakers of this morning, we are also interested into disease resistance. And for me, disease resistance, at least in open condition, we have also to follow which pathogen is infecting plants, because the pathogens are evolving too. And so to compare data from different locations is always very difficult. So now we have tools to permit to follow the pathogen evolution. And I don't know how it is considered in the shared evaluation of plant genetic resources.
Yes, so the local adaptation of pathogens is obviously an issue, or we're aware of that, and that obviously affects the different strains, and the emergence of new strains. It always affects the results that you can get. Where possible, we use either like you say artificial inoculation, so you know which strain it is, or you should do some tests to identify the strain that is prevalent in your field. And I think there's for the major pathogens, there is even database information out there. So yeah, it's definitely something that we need to take into consideration. There's like a lot of questions. Do you have time?
One minute.
One more. Okay. So there was one question on phenotypic and genotypic studies have been done on the same species in different projects. What is your opinion on correlating the results of these characterizations done in different projects with the same materials? It's a tricky one, huh? So I think really this is maybe also a good something to think about for the research infrastructure, is like, how do you create a connecting system for existing data to really make it reusable? I think the last deliverable of work package 4 is kind of dealing with trying to link existing databases. And it's difficult because every project has their own way of managing data, and it's a challenge, but I think it's an important, really an important point, and I think we should look into that. And for Frank, okay, so he wanted to know how much breeders contribute in kind, so how much that would translate into actual value in euros for their in-kind contributions by providing testing capacity. I think that depends very much on the project or on the crop, on their actual contributions. So there is, for example, in maize, we have companies that do field testing. There's companies that do some disease trials, which is a bit more intensive. There's companies that create hybrid populations. We are, I mean, it's different activities depending on expertise and capacity. And the point is always that we are generating knowledge jointly, and we also share all the information that is done within. So, thank you.
Okay, thank you very much, Sandra. Now we will shift to the last talk of the morning before coffee break, sorry. And I'm pleased to welcome Roberto Papa from Italy, and he will present phenotyping by citizen scientists in increase. And please, it's for you.
Thanks a lot, and today we'll have the chance to give some more information about the phenotyping approach and also how the experiment is working. And I'm also apologize because I have to go because I need to be at 6 for an event, so I can stay for the conference. So this is a summary on how it works, the project. So here we have an app that is increased CSA that is used for registering. Sorry, we have it because I don't have the
pointer is not.
The app basically is a true way how you register to the citizen science. And then once you are registered the app, you can sign a SNPA and receive the seeds. And the seeds are 5 bags of randomly selected lines, pure lines, already genotyped by sequencing. And also one single variety that is a Blumen variety line, single set as well. And these are very early prototypes. In this way, we have, and we also have data, very early material that are able to, say, characterize the behavior of the other. So it was pretty useful to do that. And then clearly citizens can do the phenotyping using the app and also share information, participate in Facebook group, also interact with themselves, and so on and so forth. These are what the citizens receive. They receive, as we said, the seeds of these varieties, but also a color checker. And also these are the picture that the citizens are posting in the Facebook group, but also maybe send it to us. This is what we have in the system. So we have this is the bag, the color checker, the welcome letter, and so on and so forth. And these 6 bags with this material. What is coming us now? I don't want to know how to all the stuff. One aspect is how we do the phenotyping. So citizens choose between different levels of phenotyping, and first they have to register the SSMTA, take the photo of the B lines showing that they are having received the right one, and then start to collect information by starting from choosing between different 3 levels of phenotyping.
The ECPGR is the first stage, because if you don't have the ECPGR signed, we don't send the seeds. Then the citizens are allowed also to do seed exchange through the app as well, without the citizen, always to the ECPGR, and it's a very interesting part because we have now several few thousand citizens doing that, and it is particularly useful if you want to develop a decentralized conservation as I described yesterday. Then clearly there is the validation of sowing, and with there we propose a replicated and unreplicated design, and we have them to distribute the plots on the experimental design in a very simple way, 2 replicates or not. So then as I said, the citizen have a list of traits, and they can select 3 profiles and select a certain type of group of traits that are beginner, medium, and expert. And there is no way. And in this way, we are able to have better interaction with the citizen, because at the beginning they are a little bit worried about all this work to do, and then they think that they have to do everything and everything well. So we found that with these 3 profiles, it's changing pretty well. As well, during the phenotyping work, and all the phenotype is based on this interface, these pictures, where you can make a picture, and also you can include the alphanumeric data of your phenotyping stuff. And these are the 3 different traits. So these are the traits that are the one only for the beginner. These are the additional traits for the medium, and these are the additional traits for the expert. Clearly, we are always focusing on phenotyping that are highly related to the characterization of genetic resources. Also, because one of the aims, I want to say it again, is to develop this decentralized conservation. And this phenotyping work is also a way to validate the fact that the material are stable and don't change too much in terms of phenotypic diversity. This is an example on how we use the color checker for flower color. And these are the 3 different possibility of colors. And then overall we have 1,126 lines that are from land races that are coming from 2 projects, Brezov, and that are based on snap beans. So we have both snap beans and dry beans. And BinAdapt, that was an ERACAPS project I coordinate, where we also studied the diversity of many genotypes. Again, everything is done with DOI system. And this is more or less the origin of the material, and this is the genetic structure based on the work that we recently published on the diversity of European germplasm and the relationship with the races that are grown in the US, in America. This is the material that we have here, and you see we have 107 mix between Indian and Mesoamerican, and then all the other are also assigned by races type, which is let's say sensitive to photoperiod let's say races like race Peru in the Indian group and the other one. And these are the population structure. These are the number of the material that are grown. This is an example of the bags and the amount of work that is related to that. This is the randomization unit that are based on latitude groups substantially, because there is in beans we have strong effect of day length on flowering, as is many material can be photoperiod sensitive. And these are the evolution of the group, the seeds that participate with the citizen science experiment. And I want to also to thank the Knowledge Genebank this year that they had, and then they joined in this 20 last year. And so we had a very big amount of citizens from Nordic countries. And this is very important also for the phenotypic value things. And I think that an important point is the fact that we have now more than 1,000 recurrent participants, which means that people start to work on it and they want to do it and they are engaged in the possibility of the conservation. This is decentralized conservation, which is, as I said, the point. This is some data from the phenotyping aspect. And you see that this is a flowering in relation to the control. And this, for instance, is the A2 group that is Peru, the races that is highly sensitive to photoperiod. And you see that here we have much delay in flowering, and is the round tree in the same for the same traits. This is something that we have discussed yesterday in terms of irritability and GWAS. The other aspect that I think that is important that we started with collaboration with Vito, that is a company from Belgium that do, let's say, study on climate and weather. And the interesting point is that we were able to identify a certain number of locations in Italy from which we have many information about radiation, temperature, and precipitation on kilometers per base. We select the first group of location based on the possibility to have also other citizens around. And overall, we were able to get about 500 points on which we have the phenotypic data of the citizen, and we have also considered the high quality phenotypic data and the information about the weather. So now what we are trying to do is to try to develop an understanding of the variation due to the environmental effects that could be probably able to enhance very much the possibility of extrapolating the trait from the phenotypic value, especially for technological traits like flowering. This is the information that we can get: temperature, precipitation, solar radiation, soil texture. At least for citizens, a little bit guess, because maybe some citizens don't have soil that is properly in that region. But I think it is interesting to know this information also because we have the information if they are farmer, a gardener, or let's say just people that work in the balconies. This is a first work on the best, what is 10%, 5% of successful in seed exchange. And it's interesting to see that most of the material have a specific characteristic to be Mesoamerican or Mesoamerican liture. And this can be something associated with value and the adaptability of this material, which is also related to the fact that usually Mesoamerican variety has more seeds because they have smaller seeds. So this is one possibility, that you have more seed and then it's easier to exchange. But it's also somehow related. These are the picture of the best that the citizen made. And what we see here is not only the fact that maybe they are very productive in terms of number of seeds, but also maybe the fact that are also very attractive in terms of phenotype. That among those, we have the most, let's say, in bracket, beautiful varieties. These are other citizen pictures. That is one of the major patrimony of our work. And then what we do now is we try to also train, let's say, the citizen to make pictures in a proper way. Because what we have found that even if we do tutorials and we make information for the citizen, sometimes citizens do these things with the use of color checker with the hands. And so it's not something that we can use it for artificial intelligence and for analysis. So for this reason, we try to make tutorials on how to make a picture for each trait in a way to have better picture. Like this is another example of people that put a lot of seeds even out of the. And this is our example, but I think that is very important to have also high quality picture from the citizen, because this can give us the chance to work on better information about phenotype. And these are the seeds certificate validation that is done also from our project before, because for the seed increase aspect, and then we have the original picture that we made when we multiply the material, and then we ask the citizen So sorry for a bit messy presentation. Thank you.
All right, so the question is, as a breeder researcher, what is the main difficulty to manage your phenotypic data? We can all go to the coffee break and you can think about that. It will be open for the next 20 minutes and the people online can do this also. And then we can try to see why this is not actually showing on our screen. Anyway, okay, thank you very much.